All right, good afternoon. My name is Max Bleckman, and I will be today's host of Sinobiological's May 31st edition of the Lock and Key webinar. I would like to extend a warm welcome to today's speaker, Dr. Weiyan Zhao, and to all of our viewers who are slowly joining from around the world. We love to see what country our audience members are joining from, so feel free to put it in the chat later on. Dr. Weiyan Zhao is the CEO of Aureka Biotechnologies, a fully tenured professor and entrepreneur at the Sue and Bill Gross Stem Cell Research Center, Chow Family Comprehensive Cancer Center, Department of Biomedical Engineering, and Department of Pharmaceutical Sciences at the University of California, Irvine. Dr. Zhao completed his bachelor's and master's degrees in chemistry at Shandong University, where he studied polymer, surface, and colloidal chemistry. Dr. Zhao then obtained his PhD in chemistry at McMaster University in 2008. From 2008 to 2011, Dr. Zhao was a Human Frontier Science Program postdoctoral fellow at Harvard Medical School, Brigham and Women's Hospital, and MIT. He is a recipient of MIT's Technology Review TR35 Award, awarded to the world's top 35 innovators under the age of 35, as well as the NIH Director's New Innovator Award and UCI Innovator of the Year Award. Dr. Zhao's current work with Aureka Biotechnologies is focused on high-throughput antibody discovery. On a final note, please refrain from writing any questions in the chat box to Dr. Zhao until the end of his talk. Once Dr. Zhao has finished his presentation, we will address the audience's questions. Without further ado, Dr. Zhao, please feel free to share your screen and take it away.
All right. For some reason, I cannot share my video again. Let me see. Interesting. That's okay, but you guys can see my screen well, right?
Yes, we can.
Okay, perfect. Thank you, Max, for this opportunity, and thank you, Sinobiological, for this opportunity to share our work on our channel. It's just really exciting to see people from around the world to join this webinar. Aureka is a new startup company. We're based at Irvine, Southern California, and so it just incredibly exciting. And our mission at Aureka is to transform therapeutic discovery from the conventional empirical trial and error practice to what we think the new future, AI and digital-driven rational design approach. So we do so by integrating high-throughput digital biology with AI. So we have a couple of high-throughput approaches here in terms of experimental biology. And today I will talk about this high-throughput single-cell-based functional screening. But we also have a synthetic biology-based approach for high-throughput affinity-based discovery as well. But the key here is that each experiment can produce millions of set of data, comprising a very rich data set for each of the therapeutic candidates, ranging from their genomic sequence information to phenotypic functional binding, as well as biophysical and developability as well. So our goal here is to feed all this data to our AI machine learning, deep learning algorithms, so that we can design focus of the library sequences now, and then we can go back to our wet lab technology to validate. So just because of the scale and efficiency of this iteration between wet lab and dry lab, we hope to be able to discover the design principles of the immune system, right? So we're really excited about this immunotherapy because it has been evolved for, our immune system has been evolved for hundreds of millions of years. If you think about treating infectious diseases or cancer, our immune system is probably the most powered system where we can really discover the therapeutic. So we hope to be able to uncover the design principles such that in the future, AI can just do de novo therapeutic design. So with that kind of accuracy, so that's kind of our ultimate goal. And I'll focus on bispecific antibody discovery today, but it's really a platform technology that you can apply for discovery of other, monoclonal antibodies, TCRs, ADCs, and other therapeutic modalities as well. So before I jump into bispecific antibody discovery, I thought just to kind of talk a little bit of the challenge of therapeutic discovery in general, I would see, right? So this is true for small molecule screening. This is true for antibody screening or bispecific antibody screening. I'll talk about. The field is still use kind of the trial and error approach, and that is because of limited throughput, right? So if you think about screening for a median therapeutic candidate, right? So the state of art today is still using well plates, microtiter plates, liquid handlers. You see those big labs. And for illustration purpose, if you on a screen, let's see a median drug molecule, if you're going to do 96 well plates, and you're going to need 10,000 plates, right? So that's one of the key challenge. In addition, for a lot of this screening, you typically start with the binder screening, right? So you kind of, screening for molecules that have this binding. But what we know for biologics, antibodies, bispecifics, and many of those binders do not translate the function, right? So I'll talk about a few examples. So that's why. So this is a extremely tedious process. Usually take a couple of months, and you may not get optimal drug candidate. So in our case, in the single cell high throughput functional screening technology pillar that we have, I'll talk about today, our goal is to reduce all of these processes into a single tube, where we partition millions of reactions into so this picoliter droplet, right? So this is a kind of how we miniaturize these reactions and increase significant orders of magnitude on throughput. So in addition, we incorporate this library-based approach where you can combinatorially design your library comprises many different variables. And I'll talk about that in a little bit as well. So this is really kind of significantly increase your diversity to really achieve kind of the diversity of your screening. Third, we also incorporate functional screening as well. Really address this challenge of I just mentioned, where you have to start, screen for binding first and then test if they have a function. So in our case, you can directly screen for function. So all of these features really significantly increase your discovery efficiency, right? So now you can think about some experiment that require months to do in this conventional practice. Now you can think about to do this just in a couple of days, and you also significantly reduce the cost as well, and partly because this reaction happens in this very small volume. We're talking about picoliter volumes, and the need for reagent, especially for some of the high value reagent, is very minimal. So you can significantly reduce the cost that way. But to me, the most important aspect of this is that we know screening, therapeutic screening is a numbers game, right? So it's just a statistics. So the more you can screen, the higher diversity you can have in your screening. So you can have higher likelihood to get optimal drugs in the end. So this is all of the powers of this single cell based high throughput screening. So, and with that as a introduction, now I really want to focus on bispecific, multispecific antibody discovery today. And I think everybody just super excited about this field because just for those who are not familiar with bispecific antibodies, so they're antibodies that are equipped with two binding arms that recognize two distinct epitopes or targets. It just a really incredible, exciting market, and I think it has been forecast. This bispecific antibody market is it's already like a multi-billion-dollar business each year. And if you look at these clinically approved bispecific antibodies, each of these have annual sales of hundreds of millions of dollars. So that's why farmers and biotechs are really excited about this field. So And there's have already multiple bispecific antibodies been clinically approved, US, Europe, and around the world. Just last year, if you look at these reports, just last year, there's 6 bispecific antibody are approved, and this just very significant in terms of therapeutic approval, in terms of their success rate. So this is a very significant clinical success. But also, the field has been fueled by commercial success as well, and there's been numerous of big partnership deals, IPOs, acquisition, right? So, for example, Merus, had this big partnership with Eli Lilly. Merus went public a couple of years ago. Tanel Bao was acquired by Amgen a couple of years ago as well. So these are some of the very big commercial successes, further kind of validate, the field of bispecific antibodies. So for me, what's really exciting about bispecific antibody, compared to monoclonal antibody and other forms of, combination therapy, is that bispecific antibodies really enable a very diverse range of mechanisms of actions. So this figure from this review article had a very good illustration for some of these key mechanisms, right? So I think we're very familiar with this, engaging mechanism where bispecific antibodies can bridge 2 cells together, right? So think about this T cell or NK cell. Think about this a tumor cell, and you can have this bispecific antibody to bridge these 2 cells, active, T cell, and kill this antigen-specific tumor cell, for example, right? So we have seen many of the effort in the field has been developing T cell engagers using these mechanisms. But also you can think about so this cis mechanism where you have bispecific antibody that can modulate 2 cell membrane receptors, right? So those could be 2 different epitopes from the same receptor, or 2 distinct epitopes. So you can inhibit signal pathways, you can active signal pathways. So this is a very diverse range of applications. So I will talk about example on immunocytokines, for example, or cytokine mimic antibodies. So this is just really exciting, and people also use, for example, linking to binding arms for immune checkpoint proteins, for example, right? So there's just lots of different things you can think of. So so this one is a cofactor mimetic. Think about this as a cofactor that can engage enzymes and substrate. So there's an example from Chugai for factor 8. I'll mention in a little bit. But there's similar mechanisms in the field that's really exciting. For example, there's multiple high-profile papers on PROTAPs, right? So you can think about a bispecific antibody that can engage, for example, E3 ligase, on the cell membrane to degrade, cell membrane proteins. So there's a really exciting direction going on there as well. So the last 2 panels really talk about using bispecific antibodies for therapeutic targeting to bypass biological barriers that you would normally cannot read access to the compartment, right? So for example, you can target BsABs, you can use bispecific antibody, recognize, receptors, and then you can bypass BsAB and then, deliver antibodies that can recognize, through the other arm. And so you can also do something similar to intracellular compartment as well. And oftentimes, what's really exciting here is that you can link with a toxin or drug, in the antibody drug conjugate format as well to specifically deliver drugs, to target the pathways that you're interested. So so you can see it's just really a diverse mechanism of action. It's just because there's so many different signaling pathways, so many different biomarkers, and we know in biology things happen in these multivalent, synergistic fashion, right? So when we think about so this paradigm, it just really makes a lot of sense from the biology perspective. So compared to the conventional, approaches where you target one thing, and now you can really target two or multiple things, and that's just how biology actually work to increase your efficacy and specificity, reduce your toxicity. So I think that's make lots of sense. So a lot of the effort has been focused on immune oncology using bispecific antibodies, but there's exciting new directions targeting autoimmune disorders, for example, or like infectious diseases, for example, right? So it's all based on this concept of utilizing two or multiple different pathways. So, and now I want to just talk a little bit of the bottlenecks of conventional bispecific antibody discovery. Just like what I mentioned earlier, the field is still suffer from this tedious trial and error approach, right? So if you look at a typical bispecific antibody discovery workflow, so first you need to start with binders for each panel, right? So you have to have one panel for your target A, and then you have another panel for target B, right? So you have to get those first. And then you pair them, and this many different bispecific formats. There's IgG, there's single chain, there's a VHH, there's common light chain. It's really a zoo, right? So everybody has their own kind of bispecific antibody format. We're not going to talk about that today, but just recognizing so that itself, can be a very big challenge as well. So. You form a panel of bispecific antibody construct, and then you have to individually express, purify, and then characterize them. So and just because, so this is done by conventional microtiter plates, liquid handlers. So your throughput is very low. So what that means is that it really limits the number of molecules that you can study. So that's why the field has to incorporate this trial and error approach, right? So we have to have this biased hypothesis that's we have to incorporate when we design these molecules. So for example, we might think, okay, so higher binding affinity may be better for this bispecific. So we kind of incorporate some high affinity binders, to begin with, but in reality, that may not be the case, right? So, but just, because our throughput is so limited, and this is kind of the best we can do. So that's why it's sometimes really difficult to get your optimal therapeutic candidate. So it's just a very tedious process. And, this process itself, it can easily take 6 to 9 months, just to kind of go over this process. And, you cannot even guarantee you're going to have, very optimal candidate. And this is, by the way, so this is very much dependent on your therapeutic target, your mechanism of action. So for some of the mechanisms, such as T-cell engagers, for example, CD3, CD19, things like those, right? So this approach may be okay. So you might be able to get some functional molecules, but for example, for some of the multi subunit receptor membrane agonists, for example, the vast majority of these binders may not have function, right? So, and this has become so much more challenging to get a functional candidate. So I always refer this example from Chugai. They developed this now clinically approved bispecific drug. So this is a factor 8 mimic. And it just, I really admire what they did. They used this brute force approach where they studied 40,000 individual candidates, using this approach. They have to individually express and study them. It just a very tedious process, but this is the state of art. I don't think many companies can do what Chugai did, but just to really kind of highlight the major unmet needs here in the field. So the other major challenge we kind of learned when we start to work on this field. I was trained as a bioengineer. And for engineers, so when we design something, right? So we always want to have design principles, meaning that you have a set of defined input, and you can predict exactly what the outcome is going to look like. But that's not true for bispecific antibody design. But I think that's also probably true for any other form of immunotherapeutic design. It's like the field does not have generalized design principles. Okay, so now if you look at this T-cell engager, the single chain fragment variable as example, okay, so we know that the the end function is to kind of activate T cell kill the tumor cell. But so this function depends on many, many different variables. Okay, so and that depends on the epitope of choice, the location, the distance to cell membrane, the antigen size, density, avidity, and obviously your binding affinity as well. And also just the size, the geometry, the structure, the mechanics of this molecule. We talk about the linkers, less flexibility. So, and it is clear that even a single amino acid mutation, for example, in this molecule could have a profound impact on the function. But we do not know the relationship between all these variables, right? So, and this is what I talked about, the field, why the field has to use a trial and error approach. It's like, let's hypothesize, based on the previous experience, how things work, and then we kind of design a limited amount of molecules, then study them. So that's why when you have a CD3 CD19, that may work, but when you translate that to CD3 to another tumor antigen, all of a sudden it does not work. So it's really clear that this biased low throughput approach is extremely inefficient. So we start background to tackle this bottleneck in the field, and we recently developed this high throughput single cell based approach that we call Aureka. And so let me walk you through the workflow here. In our case, we design a very large unbiased bispecific antibody library. Okay, so and I will give you show you an example where we easily incorporate tens of thousands variables so in this library design. But we make sure each cell only contains a single variant, right? So it's really make that clonal. In the meantime, so we prepare this reporter cell because, like I said, we actually do functional screening. So in one step, we're going to tell if this candidate molecule has function or not. Okay. So we use droplet macrophysics, and today, I know this is not an engineer audience, so I'm not going to go into too much about macrophysics. But if you have questions, we can talk about that after the lecture. So we use droplet macrophysics to co-encapsulate this library cells and reporter cell into this picoliter size droplets. So there's a lot of the parameters you can tune here. So for example, the size of the droplet, and you have to really think about your certain throughputs and whether or not your cells are happy with certain volume of the droplet, how many cells you want to encapsulate, for example, and all of those will determine the kind of a size of droplet you want to use. But you can easily go from 50 picoliter to 10 nanoliter, for example. So that's kind of easily tuned by your design of the macrophysics and also your flow rate. So the other key parameter here is your what we call this lambda, right? So just how many cells and the cell ratios in the droplet as well. For people who are familiar with droplet macrophysics, and so this is suffered by this intrinsic Poisson distribution, right? So if you just control your cell concentration, by cell concentration, so you're gonna get a distribution of this encapsulate cell pairs, right? So and that's typically for like a one-to-one pairs is kind of 15 to 20%, which is sufficient for most of our applications, but so that's going to be tuned as well. Okay, so that's a very important factor that you want to consider. So as I walk through, I try to kind of share the kind of lessons that we learned as well, the most important designs in this kind of experiment. So hopefully we can have some discussion later on as well. So once you encapsulate them, and then you can incubate them. So you can do this off chip. You can put this, collect this in tube and put in a incubator, or you can just do this on chip if you have temperature control and all that kind of stuff. And so this reaction typically happen very quickly. So this is we talk about just a couple of hours. And then you can have another macrophysics chip. So this is a sorting chip where you can do fluorescence detection and sorting. Okay, so I typically view this as a FACS, right? So in terms of the mechanism, it just very comparable to FACS, except that now you can sort things, based on the molecules that cells secrete, as well as cell-cell interactions, the kind of biological events that's not typically done by FACS, and now you can do this in droplet. Okay, so in our case, we have multiple lasers, so you can perform multiplexed assays. So I typically view this as a kind of a funnel, for screening, right? So we have this first point detection where we typically do enrichment, so we pick up one or two parameters that you can really do in high throughput, right? So this can be a T cell activation, this can be a BsAB assay for antibody secretion. But once you enrich them to what we call the second point detection, and this is where we have a imaging module where we can interrogate in more depth of cell morphology, for example, cell interactions, and cell metabolism, for example, so that you can not only increase your sorting fidelity, but really collect multi-model data. So that's just really great. Once you sort them out, and then you can couple with downstream NGS workflow, you can obtain the genetic information of all those sequences, or you can actually dispense live cells, you can culture them, and then you can do further high content analysis for things like cytokine secretion or tumor cell killing, metabolism. So the kind of things that may not be amenable for droplet based assay. So this is one thing I want to emphasize is that you probably hear a lot about cool technology in the field, and just every technology has its pros and cons, and just there's no single technology that can do all these wonderful things. So the cool thing about droplet is that it has extremely high throughput, but sometimes it's finicky to perform some of the biological assays in the droplet. So that's why we adopt this enrichment first, and then we have our downstream high content analysis. So this is kind of a combined approach so that we can collect a lot of different form of data. So the other thing I want to emphasize, when you do this kind of a workflow, it's just you recognize it's really an interdisciplinary effort, and so that's why we do this in-house. So we provide services to other customers. It's not something that you can just buy equipment and then you can just do it yourself, or vice versa. You cannot just have a bunch of engineers and then you just try to run this biological assays, and this really require a very close collaboration, interaction, under the same, in the same company, to develop this biological assays for a particular application with engineers, with market physics expert, with this imaging expert, all together to have, come up with a very efficient workflow. So downstream, so like I said, and you can do NGS, but you can also do Sanger sequencing. This is all dependent on how many positives that you have, and then you can do standard expression, and then you can study their functions. So and like I said, so this is very high throughput. So typically we run millions of droplet in a single experiment. That's just a couple of hours, and just because you couple with downstream NGS workflow. So that's really the cool thing about so this droplet is like you can link your sequence to function, right? So I'm not going to talk about that today, but you can also do this heavy chain light chain paired NGS. So that's kind of advantageous compared to the conventional NGS workflow, because now you have this paired information between your light chain and heavy chain. And for TCRs, you can do that for alpha chain, beta chain. So that is all kind of linked in the droplet. So that's just a very attractive feature. So, and now I want to kind of walk you through this workflow with a case study. In this case, we pick up CD3, CD19 single chain variables as example, because this is a very well established and clinically proved drug. So we thought this is a very good model system to for this proof of concept study. So like I said, first you need to have two sets of binders. In this case, we just, pick up some binders, well-known binders from the literature. For CD3, we pick up 4 different binders, and for CD19, we pick up 4 different binders. And so this panel encompasses various epitopes and binding affinities as well. So for CD19 binder, we actually performed a further sinus scan mutagenesis on the CDRH3 domain, so that we can generate a large libraries of these molecules. And then we have 4 different orientations, 4 different VH-VO orientations, and we also pick up 4 different linkers in terms of their length, rigidity, and charge. Okay, so we did this combinatorial library design, so we obtained about 22,300 different variants. So this is confirmed by NGS. Okay, so this is the key point I touched upon earlier. So by this approach, you can just incorporate all the parameters that you think important, right? So affinity, geometry, mechanics, linkers. So you can just put in. So in this unbiased fashion, so let's not presume any of those parameters has advantage or disadvantage, right? So let's just put them all in and just do the functional screening, and then you can analyze what's important, what's not important, right? So we believe this is the major advance compared to the conventional low throughput trial and error approach. So once you have this library design, and then you can engineer them into this landing pad host cell based using a HEC293 cell line. So like I mentioned before, so we have this proprietary library design and landing pad technology that allow us to efficiently incorporate each variance per cell, so that when you partition your host cell in the droplet, where you only have 1 host cell per droplet, so you can make sure it is clonal, right? So that's a really important factor here as well. So and then we could encapsulate functional reporter cells. So in this case, it's a Jurkat T-cell reporter. So the idea here is that here I should mention. So in this case, we also engineer this whole cell to express target antigen CD19 in this case. So the cool thing about this engineering approach is that you can actually tune the expression levels of your antigen, right? So that's actually really important. So of your density, avidity of your target antigen on the cell. So that's really important. So you can tune your promoter strength, and, so that you can modulate the expression level of your target. So the idea here is that if your secreted by bispecific antibody candidate has function, right? So it's going to cross-link this T-cell reporter and this antigen presenting cell, right? So it's not going to active this T-cell reporter cells. So just to be clear, in our case, because so the Jurkat cell does not express a porphyrin, so it's not going to kill the tumor cells. So it only has this fluorescent activation as a surrogate for function. So this is our assay validation very early on in the project. So before we actually do library-based screening, so we first confirm our reporter cell actually works, right? So in this study, we could encapsulate Jurkat reporter cell and CD19 positive Raji cell, and in the presence of a bispecific T-cell engager that has a sequence identical to this clinical approval drug, blinatumomab. So we can see this molecule can active the Jurkat T cell within just a couple of hours, to produce a very strong fluorescence, so that we can sort them out. Okay, so now so this is a very I would see sometimes a challenging task to engineer very good reporter cells. So oftentimes we have to spend lots of time to optimize our reporter cell. So in this case, just to note, so this is a nuclear factor of active T cell downstream of T cell activation. So we use that for this particular example. But then you can think of for any other reporter cells. This is all dependent on your target molecules that I want to interrogate in terms of your signaling pathways, right? So you can tailor your reporter cell design. So, and there's a few things you really want to consider when you want to design your reporter cells. Number one is just the intensity, right? So you want to have a very bright fluorescence. But you also want to have a very low false positives, right? So, and this is one of the key challenges for reporter cell design is that you always have this tonic, intrinsic false positive of the reporter cells. And I will share some tricks how we can address that. But you can think about like a multiplexed, design of reporter cells. So in this case, for example, we have this, signal 1 TCR, but you can incorporate, co-stimulatory activations as well to have that kind of multiplex format, so that you can be more specific in terms of the therapeutic that you want to screen. So, and here we actually screen now for that library I talked about earlier. So it's bispecific antibody library. So panel A is a representative scatter plot of your detection and sorting of the droplet, right? So again, this is very much like FACS. So we have this software that we develop in-house, but just really consider this as a FACS in terms of the mechanism. So on the left, you have this reporter cell only without this library cell. So this is your negative control. And here you notice we have 2 reporter cells, right? So this is one of the tricks that we use to reduce false positive, right? So typically for reporter cells, your tonic, this intrinsic false positive rate is somewhere between 0.1% to 1%. And when you do high throughput screening, so when you screen for millions of droplet or cells, that kind of false positive is actually a major limitation, right? So you're going to end up with a lot of false positives. So what we did here was that we incorporate 2 different reporter cells, in this orthogonal fashion, right? So the one have the green color, one have the red color. So we have those 2 different reporter cells in the same droplet. So it's only when both the reporter cells turn on, do you kind of define that as a positive. So that's actually, significantly reduce the false positives. So in this case, for example, we have a pretty low false positive rate, which is only like 0.008% in this particular example. By contrast, and now if you run your bispecific antibody library, and you now have significant positive hit, right? Because here you have your positive, right? So these are the droplet you're going to sort out. Okay, so once you sort out, if you look at the panel B, so we actually categorize a couple of the clones, and I just want to show you some of the analytical performance that we have studied about this platform. So on the x-axis, so this just a couple of the clones that we studied for this particular example. So the y-axis is abundance of those clones in the initial libraries. So this was determined by NGS. So when you design this kind of screening experiment, and there's a few quality control you could incorporate. Okay, so one is your positive control. Okay, so this is a cell line that's constitutively express this functional CD19 blinatumomab sequence. So we know that is a positive, right? So we have that positive control cell line spec'd into your library. Okay, so this is a server very good reference. So in this case, we're able to identify the positive control from this screening. So that's a very comforting. So what's really interesting, you notice this one actually, we kind of sorted out from this endogenous library, but that clone happens to encode the exact sequence of the blinatumomab. Okay, so that sequence was in the library, but we fished that out. So this is actually very assuring in terms of your discovery capabilities. Have those kind of positive controls. So these are these are 2 examples, clone 4 and clone 5. So they represent extremely low abundance. We're talking about 0.001%. So that translates to 10 copies in a million cells, right? So this is a very low abundance. So this made us think, so this approach might have this opportunity to identify very low abundance, very rare clones that would otherwise be missed out using conventional low throughput method. So once you sort the cells out, so like I said, you can go through NGS workflow if you have a lot of the positive cells. Or you can just use a very conventional Sanger sequencing, right? So you use a single cell PCR. So in our case, and then, we went, so in this particular example, we went for Sanger sequencing, and then you can do expression, and then you can study their function. So from this experiment, we recovered 265 clones, and then we studied, all of them, and we confirmed about like 80% are actually true positive, right? So our true positive rate is about 80% for this particular example. So here I show you two clones of the molecules that we identified. So they are coded in green and blue color. So red is the positive control, the clinically proved blinatumomab. So as a comparison, in terms of their ability to activate primary T cells, as well as tumor cell killing. So obviously we only studied two candidates at this point, but we already found one molecule showing this comparable property to this positive control. So what's really exciting about this technology, I think, is the ability to interrogate a large antibodies library. Okay, so now after sorting, so after you collect all these positives, and then you can really begin to study to understand the sequence structure and function relationships, right? So that would, otherwise you just cannot do through the conventional method, right? So in our case, we characterized, I think 98 molecules in this case, like a very small proof of concept. But we analyzed all these sequences, with respect to the representations of each of the variables that we incorporate in the library, right? So I mentioned that we have 4 different linkers. Those comprise, this long rigid linker and or like short flexible linkers. We have, like 4 different orientations. We have, various binding affinities for each of the binders. And now if you look at, some of this data we have collected here, and you observe some very interesting phenomena here. Okay, so if you just look at the linkers, okay, so and we found that all these 4 type of linkers, regardless their length, regardless their flexibility, rigidity, so they're all like well represented. Okay, so even this like a rigid long linkers, they work just as well as short flexible linkers. So that's kind of contradictory to the conventional wisdom of bispecific T-cell engager design. People typically think, oh, you kind of need a short flexible linker, have the 2 binders. That's kind of a more advantageous, but our data showed that, that may not be true. So orientations, okay, so you have this VO, VH, VH, VO orientation. This particular orientation that is getting significantly enriched. And, we actually, lost the other 2 orientations, and we just really don't know why that is. And it's very interesting to note. So this particular orientation is the orientation the blinatumomab adopts, right? So, and this is very interesting, and we just don't know at this point why, some of these orientations actually favor for function. Now, if you look at the binding affinity, right? So this particular clone of H38 E4 actually has the highest binding affinity, right? So and it does not really correlate to your enrichment efficiency, right? So and this is probably not a big surprise to the audience. Like, we know the higher binding affinity does not translate to a better function, right? But in the conventional trial and error design, so people try to incorporate, oh, this is the better binders, let's use that. But it turns out that may not translate to function at all. So, and just because you have this, single amino acid resolution now, and you can really ask, right? So at what side, what points does the amino acid is conserved to maintain function, right? And what are the sites that is flexible to accommodate like different mutations. This is where we studied the CDRH3, so where we did that mutagenesis of that library, right? So and here you can really start to learn the sequence function relationship at a single residue resolution. So what we were told by researchers in the field is that sometimes just a single mutation of the amino acid has a significant impact on function, and right now we just don't know why, and we don't know which one, and it just right now it just trial and error approach. So what's really exciting now is you can take all this data, right? So you can take all the design principle that we learned to train our AI algorithms here, right? So our AI models can do both predictions on the structure, but also on the bindings. And then we can use generative models to come up a defined, more defined, more focused on the library, and then we can go through this iteration multiple times and until you actually come up with your algorithm that is so accurate for any given target, any given modality. In the future, you can just do like de novo designs, right? So your AI would just design those sequences, and then you can validate. So you can kind of escape all the conventional wet lab processes. So that's really kind of the long-term vision that we have. So you can do these kind of things by this highly scalable, high throughput wet lab technologies to produce data to enable your AI design. So that was example for single chain fragment variable CD3, CD19 T-cell engager. And here I just want to very briefly touch upon another case study that we did on different modalities. So this is the full length IgG, right? So because people always ask, hey, can your technology widely applicable for many different formats of this bispecifics, because we know, there's a zoo for bispecific antibody formats, right? So, we can do single chain fragment variables, we can do VHH based bispecific, we can do, common light chains, but you can also do IgGs as well. So this is probably represent the most challenging case study that we've ever done in terms of IgGs, because we have to clone in 4 different subunits into our lightning pad cell line. So inevitably, you're going to produce a mixture of molecules, including like mispaired molecules as well. So that does represent a challenge. But so first, let me walk you through the kind of biological rationale here. And this is an example of interleukin-2 cytokine mimic antibody, right? So I use this as an example because IL-2 antibody mimic is a very attractive therapeutic field. But you can think about this for any other cytokine mimics as well, any other agonist or antagonist, so by using this principle. So there's lots of people in the field, they try to engineer IL-2 itself, right? So just give example, and there's for example, modify IL-2 with pegylation, for example, because there's one particular challenge of IL-2 receptors that so you have 3 different subunits, right? So there's alpha chain, beta chain, and gamma chain. And this is all dependent on how you engage, all this subunit. You could promote anti-tumor killing, or you could promote, autoimmune effect, right? So by promoting like Treg formation. So in order to have this anti-tumor effect, right? So people want to have molecules that can preferentially active, only the beta chain and gamma chain, and really spare the alpha chain. So people have thought about approaches to engineer IL-2 itself, with various kind of modalities, and it's just very challenging from, for example, manufacturer point of view, right? So in our case, we thought, okay, so why don't we just develop this conventional IgG, antibody format that could preferentially engage, receptor beta chain and gamma chain, right? So this will be very straightforward in terms of manufacture, right? So very similarly, we designed this library incorporating 2 sets of binders. So we have a combinatorial design again. So like I said, we clone in 4 subunits for these IgG molecules, and we also engineer reporter cells that indicative function of interleukin-2 receptor beta chain and gamma chain as well. So we do a very similar functional screening. And we have obtained a few, very promising, hits where, we currently continue to study them. So, the other challenge of, so this kind of high-throughput single-cell-based screening is that in addition to, in this IgG format where you can produce a mixture of, mispaired molecules, is that the expression of those molecules can be very heterogeneous as well, right? So, So that's could have impact on your reporter cell in terms of the molecule that you're gonna sort out. So, but the flip side of that is in our case is that it just because our library is so large, and so you have these multiple copies of the same clone in the library, and we always recommend to use kind of the cell line that you're going to use downstream to manufacture your molecules, to kind of engineer them into here, to better recapitulate, all your downstream developability studies as well. So the flip side of all the challenges that, and we kind of consider this as a one-step screening, not just for functionality of your molecule, but also your expression as well, for example, right? So, or your pairing efficiency, right? So if you have a clone that's not well expressed by this cell line, so those will be discarded, as negatives, in the sorting, right? So, and only the positive, the functional one, the properly expressed and paired ones will give you a very good signal that can be enriched. Then you can do downstream more intensive analysis of those clones. So that's kind of like a pros and cons of this kind of high throughput functional screening. So this is another example where really excited about to use this platform to discover TCRs, right? So we published a paper a few years ago in collaboration with David Baltimore's lab, using droplet for TCR discovery. But I think the principles I shared today can be really applied for bispecific engagers for TCR mimic antibodies or like soluble TCRs, like the things Immunocorn did, for soluble TCRs, for example, right? So this is a really exciting field. But so there's two things I want to emphasize, why this high throughput functional screening can be attractive for this kind of examples is that, and if you think about conventional binder-based screening, whether it's hybridoma or like display-based method where you have to first screening for bindings, right? But we know for TCR signaling, right? Peptide MHCs is that, right? So on the one hand, it's very difficult sometimes to prepare those high quality antigens, those reagent to enable this kind of a binder-based screening. But in the same time, a lot of those binders do not translate the function. So that's what we know, because nature has this, very elegantly designed TCR signaling, based on conformational epitopes of, this peptide MHCs. So this is where we believe, if you could have the opportunity to incorporate this authentic T cells in your assay to directly screen for function, so that's going to be lots of more efficient in terms of therapeutic screening here. So the other challenge we hope to address here is, for example, if you're going to screen for TCR mimic antibodies, and so you have to immunize your animals, with peptide MHCs, and they're just not a very immunogenic. You just do not have a lot of the positive B cells. So that's why, the conventional low throughput method just may not be able to get a very good clones, right? So that's true for TCR mimic antibody discovery, but that's also true for other challenging targets, such as GPCRs, for example. You just don't have a lot of the positive clones to begin with. So that's where we think the high throughput screening, where you can accommodate a much larger numbers in your starting pool, but with a much bigger diversity, you could really increase your chance to get this optimal therapeutic candidate. So the final example I want to touch upon is our partner Emberstone Biosciences used this platform, discover some very interesting CD3 antibodies. So in their case, so they were very interested in these pH-sensitive CD3 antibodies to target this acidic tumor microenvironment, to really increase the therapeutic index. So they have used this technology to discover panel of therapeutic candidate, and now they're preparing to enter IND enabling studies. So this is really exciting, but also kind of validation of this technology as well. So I want to conclude just by these slides again. So it just bispecific antibodies, multi-specific antibodies are really exciting because they really enable a very diverse range of mechanism of actions. I talked about T-cell engagers. I mentioned about you can use this tool to functional screening for TCR mimic antibodies, soluble TCRs. But we also have assay being developed to screening for bispecific ADCs, so antibody drug conjugates. So we have this very high throughput internalization assay where your antibody you not only have to recognize these two distinct epitopes on your target, but they have to be internalized in order to deliver your drugs. So we have this one assay that we can interrogate both functions. But just in general, you can view this as a platform technology where you have one cell and that can produce your library of candidate, but then you have another cell that's express target molecules that you want to interrogate, right? So and that can be any bispecific agonist or antagonist. We talk about cytokine mimics. I also mentioned early on, protaps, right? So you can use bispecific antibodies to engage E3 ligase to degrade a protein of interest. But you can also target, multi-pass, cell membranes, such as GPCRs. Some of the very challenging target that you are not easily able to do using conventional methods. So we're a startup company, so we're proactively seek collaborations and partnerships with farmers and biotechs. So if this is something you're interested in, I would very love to speak to you afterwards. Okay, so I think I'll stop here and let's see if there's any questions.
All right, Dr. Zhao. Andy Liu asks on slide number 9, why HEK 293? Any concern with other cell lines like CHO? Just curious.
No, you can do true cells as well. Yeah.
Understood. In the next question is, what is the expression level of each hit in the single droplet?
Yeah, the expression level of each of those molecules. So in the example I showed today, so we actually did not really consider that in the assay development. So we just sort out the positive clones that can active the reporter cells. It's kind of a yes or no, like a binary kind of assay. But we have previously incorporated, for example, like a BsAB binding assay in the same droplet, where you can actually quantify the secretion level of each particular candidate as well. If you need to calibrate expression levels, you can do that too.
Understood. And then Betty asks if it would be possible for you to talk a little bit more about the picoliter droplet technology.
Yeah, so so that is a in-house technology, and which is based on droplet macrophysics. So I mean, right now we use a flow focusing mechanism to form droplets, so like I said, you can tune the size of droplets. But so we have had a couple of innovations that we did in that workflow. For example, one challenge in detecting cells or bees in that droplet is a focus problem, right? So how can you focus on that cell within that droplet so that you can ensure your sensitivity? So we actually incorporate a dual focus laser system where we can have multiple focus points in that droplet. That's really kind of increase our sensitivity. So that's just one innovation that we did. And the second thing I want to mention is that we have this multi-step imaging system, right? So the first point is enrichment, the second point is imaging, so we can collect different form of data as well. So those are some of the differences that we have built in-house compared to some other macrophysics in the field.
Understood. That appears to be the last question. So Dr. Zhao, I'd like to thank you for giving a very interesting sneak peek into some of the technology and methodology that Aureka is utilizing. I'd also like to thank our audience members. We've had people join from Kuwait, Malaysia, Germany, as well as the United States. And if you have another second of time, we actually just got another question. It's about the picoliter droplet technology and whether it can be any different from the beacon.
The advantage of the droplet is the throughput, right? So you have I would say two orders of magnitude of higher throughput than the beacon technology. And that's really important because for a lot of applications where you start with lots of different cells, right? So if your diversity is very high, you have to start with a bigger library, or you have to, start with, B cells, for example, from, humanized rat, or where you just have lots of cells. Let's see, you have lots of cells on a screen. So the droplet is advantageous because of the high throughput. Yeah.
Let's see. Well, on that note, we will conclude the webinar. If anyone is interested, this webinar will go live on YouTube later today. So look out for that link. And Dr. Zhao, thank you very much and have a nice rest of your day.
Thank you, Max, and thank you, everyone.